Synthesizing proteins and regenerating cells. Proteins are very important for the cell and are essential to a functioning life. But how are proteins naturally synthesized within our body? Our body has a blueprint for each protein in a codified form, which is stored in a molecule called deoxyribonucleic acid, better known as DNA. Generally found in the cell nucleus, our DNA consists of bases with different characteristics: adenine, guanine, cytosine, and thymine. The DNA is like a vast library with volumes of encrypted books, and its counterpart, ribonucleic acid, or RNA, acts as the Rosetta Stone. The RNA has the ability to recognize the code within your DNA, and through this process, it builds the proteins your body needs, each with its own structure and functions. As research and applications progress, protein and peptide therapies started evolving as biochemists began to understand the molecular structures that produce the desired responses within the cells. These very small molecules are the crucial messaging element of the cell and can contain the sequence of information needed to stimulate a response within damaged tissues. Refining our treatments down to the molecular level eliminates additional genetic material that hinders stem cell treatments and other cellular therapies. This unnecessary extra material can include molecules that can make the treatment less effective and even harm the body. Not only does this allow for natural and safer treatments, but it also allows for more precise targeting within the body. The proteins and peptides are formed from amino acids by covalent bonds between adjacent bases. As the number of amino acids increases, the chain gradually increases its length and a new protein is created. The final sequence of amino acids results in one protein, which is coded with the functions to produce specific responses within the cell. The chemical message within this protein is then delivered to the cell through various signal transduction pathways. Cells are capable of responding to a variety of different stresses or signals by increasing or decreasing the availability of specific proteins. The complex cascades of events that lead to changes in the expression of specific genes are known as cell signaling pathways or signal transduction pathways. These pathways regulate numerous cell processes, including growth, proliferation, and death or apoptosis. Signal transduction pathways. Signal transduction pathways are very important for cellular actions, but they are sometimes misunderstood. Consider this analogy. When you strum an electric guitar and vibrate the strings, it passes the sound signals to magnets and wires, which transduces that message into an electrical signal. The amplifier then picks up this signal so you can hear it at higher levels. In our bodies, transduction occurs when cells are switching a signal message received from outside the cell to a message for specific actions within the cell. It starts with the introduction of a chemical message finding a receptor within the cell membrane. A receptor is a binding site on the extracellular domain, which specifically recognizes the signal molecule referred to as the ligand. The interaction of the ligand and the receptor alters the tertiary or quaternary structure of the receptor, including the intracellular domain. These structural changes are not sufficient to yield an appropriate response because they are restricted to a small number of receptor molecules in the cell membrane. The information embodied by the presence of the ligand, often called the primary messenger, must be transduced into other forms that can alter the biochemistry of the cell. Changes in the concentration of small molecules, called second messengers, constitute the next step in the molecular information circuit. Second messengers will begin to relay information from the receptor-ligand complex. Particularly important second messengers include cyclic AMP and cyclic GMP, calcium ion, inositol, 1,4,5-trisphosphate, IP3, and diacylglycerol, or DAG. These secondary messengers are going to spread throughout the cell and target the protein kinases. The protein kinases are made of a number of different subunits of proteins. It consists of 2 catalytic subunits, which speed up chemical reaction, as well as 2 regulatory subunits. As long as the regulatory portions are attached to the catalytic portions, the protein kinase is inactive. Secondary messengers will bind to the regulatory subunits, releasing the catalytic subunits. Phosphorylation then occurs by ATP, or energy being added to the catalytic portions, activating them. Protein transcription. Once the catalytic subunits of the cell's protein kinase is activated, the coded message can pass through the nucleus of the cell until it arrives and stimulates the DNA. When this process is complete, the RNA reads the new information and creates a functional protein based on the needs of that particular cell. This causes the cell to start functioning normally again. Now, let's take a closer look at how this process takes place in your body billions of times each day. The DNA double helix within all humans contains 2 linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information needed to create the protein. The double helix is unzipped and one of the strands is copied. The strand that is produced as the DNA is unzipped is called RNA, which is a close chemical cousin of DNA. The building blocks to create the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the gene. At this point, the RNA needs to be edited through a process called splicing before it can be translated into a protein. The spliceosome removes all the introns, so that the edited RNA only includes exons, which are the complete instructions for the protein. When the RNA is complete, it snakes out into the outer part of the cell. Next, all the components of a molecular factory, called a ribosome, lock together around the RNA. It, then, translates the genetic information within the RNA into a string of amino acids that will become a protein. Special transfer molecules for each of the 20 amino acids brings each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read from the RNA, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. The amino acid is added to the growing chain, and within a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends on what genetic message you feed into the RNA. Supplemental protocols. These additional molecules are used in conjunction with RCT's treatments, depending on the presence of key factors for each patient. Effects of polyphenols on cell signaling. RCT's treatment protocol for autoimmune diseases and other conditions involving inflammation within the body includes polyphenols, which are a group of chemicals found in plants. They are characterized by the presence of more than one phenol group per molecule, or ring-shaped chemical closed structure. They are mostly found in red wine, green tea, coffee, and chocolate. Some of its main functions are reducing the risk of chronic and degenerative diseases, such as cardiovascular disease, because they inhibit the oxidation of low-density lipoprotein, or LDL. Additionally, they are potential agents that reduce the risk of cataract formation in diabetic patients. Polyphenols are the most important antioxidant phytochemicals found in vegetables and fruits due to biophysicochemical properties. They are potentially beneficial to health because they are molecules with anti-mutagenic, anti-diabetic, anti-inflammatory, antibacterial, and anti-tumor activity. Some reports indicate that they also inhibit HIV. Polyphenols are currently studied for their diverse pharmacological actions on the treatment of various diseases or phytochemical therapies, and are currently considered to be life extenders. Depending on the condition being treated, RCT uses a specific polyphenols to boost the effectiveness of its treatment protocol. Flavonoids, which are in the family of polyphenols, are characterized chemically by two benzene rings joined by a linear carbon chain. Flavonoids are commonly defined as dietary antioxidants for inflammation. However, many of the biological effects of flavonoids appear to be related to their ability to modulate cell signaling pathways. Evidence from studies indicates that regular consumption of foods containing flavonoids may reduce the risk of several chronic conditions, including neurodegenerative diseases, atherosclerosis, and certain forms of cancer. The considerable results prompted further research into flavonoids' ability to affect chronic disease by regulating numerous cell processes through signal transduction pathways. Effective signal transduction requires proteins known as kinases, as mentioned earlier, which catalyze the phosphorylation of target proteins at specific sites. Flavonoids can selectively inhibit kinases that initiate cell signaling cascades by binding to specific receptors in cell membranes, helping to regulate cell proliferation, inflammation, invasion, metastasis, and activation of apoptosis. Flavonoids are an addition to the protocol when RCT treats patients with autoimmune disorders, such as diabetes and rheumatoid arthritis, as well as other conditions where inflammation is an issue. Antimicrobial peptides. In the last decades, the indiscriminate use of conventional antibiotics has generated high rates of microbial resistance. This situation has increased the need for obtaining new antimicrobial compounds against infectious diseases. Among these, antimicrobial peptides, or AMPs, have proven to be effective against various pathogenic microbes. Additionally, AMPs are usually produced by a non-specific innate immune response and play a critical role by directly combating susceptible pathogens and by recruiting and activating leukocytes at sites of infection. These antimicrobial peptides are involved in the inhibition of cell growth and in the killing of several microorganisms, such as bacteria, fungi, enveloped viruses, protozoans, and other parasites. These therapeutic agents can be isolated from different organisms, being widespread in nature and synthesized by microorganisms, plants and animals, both invertebrates and vertebrates. They have many properties similar to antibiotics, such as relatively small sizes, below 25 to 30 kDa, amphipathic structures, cationic nature, and offer low probability for the generation of microbial resistance.